In this video, we are going to examine the process of how cells divide, with a particular focus on the important role played by structures known as chromosomes. All multicellular eukaryotic organisms, whether they are humans, plants, or even large animals like walruses, require a constant supply of new cells to support their growth, development, and the repair of damaged or aging tissues. This need for new cells is especially critical in young organisms that are actively increasing in size, but it is also essential for older organisms who need to continuously replace worn-out cells. A good example is the skin, where cells are constantly shed and replaced as part of a healthy maintenance cycle. The biological process responsible for creating new cells is known as the cell cycle, and it can be thought of as the complete life story of a single cell, beginning when it is first formed and ending when it divides into two genetically identical daughter cells. There are three main stages in the cell cycle that you need to understand. These stages are growth, during which the cell increases in size and produces more subcellular components such as mitochondria and ribosomes, then DNA replication, where the genetic material is duplicated so that both resulting cells will receive a complete copy, and finally the actual division phase, which includes mitosis followed by cytokinesis. During the initial growth phase, the cell enlarges and builds more structures it needs for proper function and replication. Then, in preparation for division, the DNA is copied in full so that there is a complete genetic set available for each of the new cells. To fully grasp this, it helps to understand how DNA is organized inside cells. Normally, when a cell is not actively dividing and is simply performing its everyday functions, the DNA is found in long, thin strands spread throughout the nucleus. However, as the cell prepares for division, this DNA becomes tightly coiled and forms structures called chromosomes, which are compact packages of genetic material. Each chromosome contains a vast number of genes, which are specific sequences of DNA that code for particular proteins and control different traits like eye color or blood type. In eukaryotic cells, such as those found in animals and humans, each chromosome is present in two copies, which together form a homologous pair. One copy is inherited from the mother and the other from the father. Humans have 23 pairs of chromosomes, making a total of 46 chromosomes per cell, although different species have different chromosome counts, which is one of the reasons why animals from different species cannot reproduce together to form fertile offspring. As the cell gets ready to divide, each of the 46 chromosomes is copied, but instead of separating immediately, the duplicate remains attached to its original, forming an X-shaped structure. Each half of this chromosome is identical to the other, and each side is often called a chromatid or arm. The DNA sequence on the left arm is exactly the same as the sequence on the right arm. When the cell enters the next stage of mitosis, all of the chromosomes align themselves along the center of the cell in a straight line. Protein fibers called spindle fibers extend from opposite sides of the cell and attach to each chromatid or arm of the chromosomes. These fibers then pull the chromatids apart, moving them toward opposite ends of the cell, which are called the poles. This pulling action splits each chromosome into two identical halves so that each side of the cell now contains an exact copy of the genetic material. This precise separation ensures that both resulting cells will be genetically identical. Once the chromosomes have been separated and moved to opposite sides, the final phase of the cell cycle begins, which is known as cytokinesis. During cytokinesis, the cell membrane begins to pinch inward, and the cytoplasm is divided between the two new cells. The result is the formation of two completely separate daughter cells, each with its own nucleus and full set of chromosomes. In humans, this means each new cell has 46 chromosomes, just like the original parent cell. These new cells are genetically identical to each other and to the original cell they came from. They can now go on to perform their functions, or, if needed, they can enter the cell cycle again and continue dividing to contribute to tissue growth, repair, and development throughout the body.